Now more than ever, innovative technologies are fueling change and sparking new ways of thinking. Collaboration between corporations and startups is key to staying at the forefront of these trends. However, finding the right startups can be expensive, time-consuming, and ineffective. But Plug and Play is here to help. As a corporate partner, you will gain access to a whole ecosystem of innovation. Discover startups that meet your tech interests. Stay updated on the latest trends and network with industry peers. We will help you during every stage of your innovation journey, no matter where you are and where you want to go. Get in touch today.
All right. Good morning, everyone. Welcome to the Hyper Hope 20 Years of Precision Medicine webinar hosted by the Plug and Play Health team. Just give everyone a couple moments to trickle in here and go through a few housekeeping items before we really kick things off here. So just briefly, an overview of today's agenda. We'll start things off with a fireside chat with Dr. Francois Bernier, a leader in the implementation of genomics and precision medicine approaches in the Canadian healthcare system. We'll follow this up by 3 startup pitches from companies that have built on the decades of technological advancements of genomics and are really forging new application areas in improving health outcomes. So just a few housekeeping items. We do encourage you to submit questions both for the fireside chat section and for each of the startup pitches. There's a Q&A function down at the bottom of the window. So if you enter your question there, the moderator will be able to ask that question to the speakers. Please use the chat feature for any general questions or comments to the Plug and Play team or the rest of the audience. And then, of course, goes without saying these days, but please remain muted unless you are presenting yourself. So before we hop into the chat, I'd like to give a brief introduction on Plug and Play itself. So Plug and Play is the world's largest innovation platform and one of the most active VCs in Silicon Valley. We catalyze technological advancement by connecting corporations with the brightest startups across the world. We're actively supporting innovation in almost every major industry with a truly global network of partnerships. In healthcare specifically, we focus on digital health technologies that challenge clinical practices, improve data analytics, and enhance the patient journey across the continuum of care. Our platform has 3 major pillars to it: startup acceleration, corporate innovation, and venture investment. If you want to chat more or learn a little bit more about Plug and Play itself, please feel free to reach out to me after today's webinar. Just for some background, I'd really like to set the stage for our discussion today and sort of provide an update on where we are in the evolution of precision medicine. So it's really been 20 years since the first draft of the human genome was published, from decades of collaborative research and dedicated investment on a scale reminiscent of the moon landing. Since then, the industry has moved at a breakneck pace in technological advancement, along with it the promise of new generation of medicine guided by a deep understanding of the genetic underpinning of every disease. Now, whether it was Barack Obama's declaration of the Precision Medicine Initiative in 2015, to the revolutionary discovery of the CRISPR gene editing techniques, the industry has had its fair share of hype and time in the limelight. Now, more recently in 2022, a team of scientists from the Telomere to Telomere Consortium have confidently sequenced and published the remaining 8% of the human genome map that remained elusive for many years. Breakthroughs in long-read sequencing technologies from companies like Pacific Biosciences and Oxford Nanopore gave scientists the tools they needed to sequence these highly repetitive and tightly packed regions of the genome. Not only did this yield the first gapless human reference genome, the team actually provided new insight on over 600 genes that are linked to human health and disease. Now, as we enter the 3rd decade of precision medicine story, we're delighted to have a number of very special guests with us today who will be sharing their unique perspectives on implementing genomics and precision medicine in the 21st century. So with that, I'd like to introduce our first guest and welcome him to the stage. Dr. Francois Bernier is Professor of Medical Genetics and Pediatrics at the Cummings School of Medicine at the University of Calgary. He's also head of the Department of Medical Genetics and Section Chief of Clinical Genetics within Alberta Health Services. The stack of hats is quite high, as he also sits on a number of national efforts to bring whole genome sequencing into the standard of care. Welcome, Francois.
Hi, Niall. Good to be here.
Fantastic. Thanks so much for joining us this morning. So maybe just a kickoff question for you here for our audience. As a practicing clinical geneticist and academic researcher, I'm wondering if you could describe for our audience really the kinds of patients that you see through your clinic on a daily basis.
Yeah, so maybe I'll describe it sort of clinic-wide. We have really a full scope of service and pretty typical of genetics these days. So we'll see couples who come in thinking about starting a family, so thinking about their risks either because of personal history or family history. We have a large prenatal practice, so all kinds of things coming up or arising during a pregnancy that may be complex. And the bulk of our work is pediatric genetic and metabolic diseases, so the hard stuff that hits kids early and creates complex illness. And so that's the diagnosis treatment part of pediatric disease. And probably our biggest growth of medicine actually over the past, particularly 10 years, has been the adult side. So cancer genetics, neurogenetics, nephrogenetics, cardiovascular genetics is kind of, we found genes for a lot of the adult onset chronic diseases. So my personal practice spans quite a bit of that, but certainly our whole clinic spans all of it.
Fantastic. And you've seen quite a few patients over the years and the evolution of the tools that you use in your clinic on a regular basis. And if you think back to your days in medical school and to what you can achieve with patients today, I mean, what would you say has been the most impactful technological sort of change in the field of genetics and genomics that you've been able to realize in your practice?
Yeah, it's a good one. It's changed a lot, right? So when I started, we had about 20 or 30 genes. They were hard to test. And that's kind of the extent of our knowledge. And I knew them all, right? I felt I was really smart, is there a gene for that? I'd be like, yeah, we know that one. But really it's this thing called massively parallel sequencing or next generation sequencing that's completely revolutionized the field. So probably about 20 years ago, instruments started showing up on market. Initially in the research field, and for about 15 years have really been heavily implemented in research and clinical practice. And these guys sequence not only just little fragments one at a time, it can do millions of little fragments of DNA. And so the next generation sequencers, PacBio, Illumina, Oxford Nanopore, et cetera, all of them can allow you to do things that were just, science fiction-y, right? So for the price of about $1,000, now you do a human genome. That was a $200 million sequencing project about 20 years ago. So you unleash that technology in the hand of scientists, innovators, and clinicians. And now you can sequence tumors, you can do whole genomes, you can do genomes of pretty much any organism. You can look at the expression of a single cell, what is genes are turned on or turned off in a tumor cell or an immune cell or a host cell. You can sequence bugs. That's how we got the COVID genome so quickly. You just threw it on these sequencers. So yeah, by far, if you look at technology change, maybe globally, if you look at technology change, the arrival of these next generation instruments was just really transformational.
No kidding. And just the resolution you can get to in all these different fields. And I know it was one of the key tools in our toolbox in trying to keep ahead of the COVID-19 crisis, being able to understand the genome evolution and be able to ideally, fine-tune our vaccines to keep up with the evolution of those viruses. You spoke to sort of how in the early days it was a $200 million effort to get the first genome done, and now it's close to $1,000, if not a little bit lower. With that sort of transition to democratizing the availability of genome sequencing, there was naturally a lot of hype and optimism that we'd really be entering a new generation of medicine as these tools became available in every single aspect of human health. At the turn of the decade in 2010, those expectations were a little bit dampened as we kind of realized maybe human biology is quite complex. Lots of diseases contain many gene implications and genes at play in them. So I think some of those expectations and some of the hype has been tampered a little bit. In sort of your practice, in your perspective, in what ways do you think it has lived up to the original promise and the original hype when these technologies came available?
Yeah, it took a while, right? You know, the human genome, there's probably a gap of 10, 15, 20 years after the genome was completed. And it took technology change, but it took knowledge to advance. You know, we just had to sequence huge amounts in my field, you know, of patients, but also norm, like just understanding normal variation. And this is a place where we haven't democratized enough. You know, I don't understand genome variation in some of our, you know, think of the diversity of the African populations. You know, if I have patients who come from an African, Black African descent, I don't understand their normal variation, or indigenous populations, et cetera. So I think we just had to understand what was normal variation. So what does a typical human have? But we also had to understand diseases. But it's really hit now. You know, I routinely use next generation sequencing technologies in my practice. I have a diagnosis rate of, you know, our clinic overall, probably 40, 50, 60%. We're not hitting that 70, 80%. Maybe the rest of them are genetic anyways, but, you know, it used to be 5, 10, you know, and so that ability to rapidly arrive at explanations, and some of it's driven therapy, some of it hasn't, but it's definitely here. And in most, you know, clinics, is it available to everyone? I don't know. I think that's globally, that's a huge challenge. It certainly isn't. But we're getting there, I think.
For sure. No, that's a fantastic point to bring up. Maybe following on that a little bit, just around sort of the access and healthcare equity. You know, healthcare and equity in general has something that's become all too apparent to us, especially since the COVID-19 pandemic exposed some large cracks and gaps in our system with regards to the health outcomes as it's related to social determinants of health. Really, I mean, when it comes to genomics and precision medicine, how do you feel from sort of an academic research perspective that the research community should play a role to ensure that the future is definitely a genomics for all future, regardless of race, ethnicity, or other factors?
Yeah, I think that we need to, right? And the way to do it is to not silo our genome data. You know, I think we have to understand that genome data is a legacy. It's our human legacy in many ways. And how do we make that data more available in a consistent manner? So, but also linkable. So a lot of our work now is shifting towards not thinking of genomics in isolation and precision health as being a pure omic field, but thinking of precision health as trying to deeply understand those individual biomedical and social determinants that in the end combine to result in a risk of disease. So how do we focus on prevention, early diagnosis, and then trajectory? You know, are you going to have good outcome, poor outcome? And then how do we develop targeted approaches to early stage interventions? Think of early cancer interventions. You know, how do we stratify earlier? And then those really high tech things, but they all have to be kind of pushing that cost boundary down. We need to think of innovation space of driving these costs down that make them affordable. And that's how you get equitable access, is by developing solutions that are affordable. And I think, you know, certainly our approach to precision health and genomics has shifted a lot to include the socioeconomic determinants of health and linking our data. So how do we link genome data to those data sets to start understanding, you know, how are all these combining to result in individuals who are at increased risk or not? So I think that's one approach to democratizing. I think you need a conscious effort too. Like, you know, you need to fund projects and identify projects that support accessibility of technologies to equity deserving groups across the spectrum. I think we have to share our wealth and resources and knowledges. Like, you don't have to have driven every version of the Pinto to buy a next generation sequencer. You know, you can jump into the technology now, but how do we support individuals doing that? And particularly on a global community scale. And I saw a beautiful map a while ago of sort of where NGS instruments are located. You know, they're not located equitably across the world.
Yeah.
And that kind of makes sense intuitively, but it's sad to see, right? So, and so I think that you need a conscious effort to try and do this. And I think the funding agencies are pushing us. Like, we just submitted a large grant, and boy, if you aren't addressing how your approaches were going to address issues of access to equity deserving groups and addressing the barriers, you aren't going to get very far. So I think the funding agencies are going to play a role here, and lots of challenges in the system, around equitable access, et cetera. But
yeah, for sure. Maybe we want to drill a little bit deeper into that. So obviously technological advancement and reducing costs, and making a technology that can be deployed in sort of a leapfrog manner to these areas of the world where maybe they didn't have the original sort of evolution of Illumina sequencers. So technology advancement is of course an important factor, but the other side of the coin is kind of governance and policy issues all around providing better access and making sure that these underserved communities do have access to this next generation of medicine. I mean, I know you play a role on a number of sort of steering committees nationally that are really trying to change the policy side of the equation to make sure that these technologies are implemented safely and effectively. If you were to pinpoint maybe one or two things that policymakers really should be focusing on, either in Canada or kind of internationally, to help really drive forward equitable access, what would you say those would be?
Yeah, I think there's a really techie part of it, which is just standards. So how do we standardize nomenclature and data? So health data is not standardized. Genome data is getting better. So there are global standards now around how, what type of format your data should be in so that it's shareable and readable by other individuals. But it's the policy side, right? And I really don't envy some of the policymakers because they struggle between the challenge of particularly protecting individual privacy, particularly when we look at health data. So, we're a federation of health systems in Canada. So, in Alberta, the data is held by the Alberta government and governed by the Alberta government. And I can do a lot within the province. We have amazing data sets, but how do we share that? So understanding how to continue to protect privacy versus also recognizing that there's good here, and how do we inform individuals of the use of their genome data, particularly linked to health data. Commercialization is a good one. I think we should think about, how do we bring the value of commercialization of some of this and return some of that value to healthcare systems or to patients, if that's going to be a route. So the policy side would be really, it's challenging, and we're trying to share data across the country. And I can tell you most of it is around this tension of, open science sounds great, and it's going to share absolutely everything. Or in the healthcare system, we're just going to share and de-identify, but is genome data de-identifiable, et cetera. So we need stronger policies. Canada does have a digital health strategy right now that is looking at this exact question of how do we share, and then I think we need to do the same globally. None of us, particularly in genomics, can solve these issues in isolation. So we need larger and larger data sets. You're going to hear a little bit about machine learning approaches and AI learning approaches to data. We need high quality data, and we have it.
Exactly.
We just have to make it accessible.
Yeah, for sure. I mean, that population scale genomics is really where a lot of the advancements in understanding human disease are really going to come from, right? And we have the data sets. We just need to find the appropriate mechanism ways to put them together to fully leverage the value. So it's clear, genomics has played, a game-changing role in your ability to diagnose patients that come through your clinic. You're saying you're upwards of 40, 50%. That was probably unheard of, 20 years ago. Patients with rare diseases coming through, getting an actual identifiable cause for their symptoms. Further down the sort of care pathway, how has the availability of genomics and maybe gene editing technologies, how has that improved the treatment options that are actually available to you as a clinician to these patients after they get that diagnosis?
Yeah, not surprising. A little bit of a lagging space, right? So, but it's coming. We have access now to specific gene therapies. So we do have gene therapy for a number of genetic diseases that have been approved and are in clinical trial. We treated a patient just recently with one of these. And that was really been the curve. If you want to look at cost effectiveness, the cost on the healthcare system, we look at viability of our healthcare systems, is heavily driven by a very small percentage of patients who have very, very complex healthcare needs. And those needs costs actually spread out heavily to the patients in society. Loss of income, loss of productivity. And so we have some specific therapies. We often can redirect therapy. So, redirect what we have to screen for, et cetera. We've had more impact on the adult side, I would say, where, most of the disease conditions are screening type of conditions. So think of hereditary breast cancer, colon cancer, cardiovascular disease. Like those can have huge impacts because you identify people at risk and you tailor their screening, early identification of tumors. We've had some really amazing successes in that space. And now we're sort of in this, there's going to be a wave, and we're starting to see it of clinical trials coming through for specific therapies, whether they're mRNA therapies driven off of the COVID world, gene editing technologies coming in, specific molecules being developed. Cancer is a leading edge here. Pharmacogenomics is coming in, so testing for people who would have better responses or avoiding adverse events from drug therapy. So pharmacogenomics is coming along, therapies for rare diseases coming along. Like we're seeing those leading edge areas that were the first adopters of the genome technologies really starting to drive into therapy space now.
Yeah, that's fantastic. And I think that's only going to grow as the technologies become more widely available. I feel like we're kind of at the early 2000s era of the new generation of medicines as we were with genomics at the time.
Yeah. I mean, there's always those great examples. We have kids who are chronically in hospital, very sick, and you throw them on sequencer and you get an answer in a week, and you're like, whoa, like these kids need to be transplanted, and they're cured by their transplant, right? Whereas fussing around, not sure what they were doing, you were risking their life really. And so we've had some of those successes. Most of the places working in this space have seen those.
Yeah.
I think the other one that seems to be changing a little bit is the concept of polygenic risk scores. So, are there little variants that we have in our genome that put us at risk of disease? And we realize it's not all those rare diseases, but, we know these things run in families and why. And that could have significant impact if we could stratify people into, high risk cardiovascular disease risk. And you could modify that using standard therapy or targeted therapy. So I think that might be another wave of more generalized application of genomics. Into more population level prevention.
Yeah, for sure. I want to maybe pivot a little bit for our last sort of few minutes here and talk about something I know you're very passionate about, but from much of the population, they've learned the terms genetics and genomics from their 23andMe kit, maybe that they get mailed to them, and sort of that consumer-facing genome sequencing products. Now, it's certainly a positive that genomics and genetics are part of the household, like dialogue and conversation, much more than they have been in the past. But I still feel there's an element of maybe confusion out there what those kinds of tests are really able to accurately and confidently sort of deliver when it comes to medically actionable information. I'm wondering, in your clinic, you see a lot of parents, you obviously see a lot of adult patients as well. How has the sort of genomic literacy maybe improved over the years, and has that been helpful to you when you're kind of explaining, the use of genomics in their care pathway?
Yeah, there's definitely early adopters out there, right? So, and yeah, we see them. So for sure, either they themselves have undergone kind of consumer-based genomic testing. What I've seen mostly though is people becoming much more aware and connected as a community. So, they come in with a much greater understanding, not all, but, the early adopters come in with a much greater understanding of genomics. They kind of come in knowing what I'm going to offer them, and they've already read about it and they want it. Some of them want access to their genome data. Some will do their own genome data mining. It's an interesting question, right? So do you hand off their clinical genome and let them go digging around? But so there's early adopters out there. It's improved. I think it's just complicated still, right? And understanding variants of unknown significance, these gene variants that we're not sure whether they're real or not, it's still really hard for most people. Understanding negative tests, the lack of predictability sometimes of tests, right? So, you have a variant and it causes a 60% risk of breast cancer. Well, that's a tough concept, at 60% at what age, and is that yes or no? Am I high risk, low risk? So it's still complicated out there, but literacy is improving, and that's a good thing, right? We want good literacy around this stuff, so people can make good choices about the use of genomic information for their personal health, their family health, particularly their kids' health, right? And there's tons of marketing, which is great. I have nothing against, good use of genomic technologies, but it's complicated.
For sure. No, thanks for that. It's we'll get into some questions just from the audience here. But maybe one final one for you, Francois, for myself here. I mean, looking forward, the next decade of your practice and precision medicine genomics. I mean, what excites you the most when you look at the pipeline of technologies that might be available to you in 2032? What gets you excited the most in your ability to impact future patients?
Yeah, I couldn't predict where we were today, right? So I think we're going to see another bump in technology. I mean, I think we need another drop, right? So if you go from $1,000 genome or $1,000 single cell sequencing test to $100 or $10, if that drops, then you're going to see a whole other level of access. And then pretty much we've democratized it. It's no longer the cost. I'm worried about the interpretation of it and the good quality reinterpretation of genome data and how people will hold that. And I know one of the companies we're going to hear from today is working in this space of, how do you help good interpretation, reinterpretation of genomic data, and then therapies. I think we're just going to see this explosion of targeted use, I hope, of this information, not just telling you your risk, but modifying your risk. But yeah.
For sure. So just a couple questions from the audience here, Francois, for you to answer. So back on the topic of sort of consumer-facing, I guess, genomics products. So the question asker is saying, you imagine that a number of patients show up in your clinic with a test result from 23andMe in hand, with a number of sort of questions and concerns around it. Realizing the limitations of these products that these tests provide, how do you as a geneticist deal with this? And then follow up to that is, do you see a role or value for genomics-based private payer consumer tests in a publicly funded healthcare system in Canada?
Yeah, so two really good questions. So the way we currently deal with the 23andMe or direct-to-consumer test results is, if that individual has a genetic change that is in one of the genes that we would be testing for and we think is actionable. So if someone shows up and says, hey, I have a breast cancer mutation, and they show us their report, and we're like, okay, yeah, that probably is real. We will see them clinically, no different than if they had been sequenced in another province or somewhere else. What we won't do with them is we just don't have the resources to get into all the other things that 23andMe tries to promote, good or bad, because we just don't understand their data set. You have a multifactorial risk of breast cancer or colon cancer, or your pharmacogenomic type things like, or someone wants to come in and discuss their ancestry testing. So, there are limits, but if you have, you're a carrier of a rare disease, regardless of how you found out, we will help you out. If you have a known genetic disease, we will validate it. But it's been pretty limited, actually. And that's because, most of these tests aren't going to identify those things. The second one, is there a role? I think, this is, I work very closely with an amazing health economist, and she's taught me a lot about value, right? So I think until we understand in the healthcare system whether there is adequate value for a test, any intervention, and we have to be value-based. Are we getting enough value for healthcare dollars to be spent on something? I think there is room for consumer pay or whatever you want. I think the healthcare system is stressed. I think it has a lot of priorities. And so we're also spending a lot of time to collect that value data, right? If we want genomics to be part of the healthcare system, you can't show up and just say this is important. You have to say this is important because it has value, and value is just not decreasing costs in the healthcare system. Is value to quality of life, decreasing cost, productivity, like the broader statements of health. And so I think if genomics wants to play in the healthcare game, you have to go up against the cardiologists who are saying, we want a new fancy, treatment for coronary artery disease, and we have all the data that would show this can be cost effective, and we have to show up at the table with the same data, right? So short answer, sure, until I think we can prove value to the system. We've been able to do that for rare
Yeah.
disease diagnostics, but there's going to be tons of applications. So same thing, venture capitalists are thinking, we want to develop a test. And how do we get the healthcare system to adopt it is a really, really important question. So a line of sight of, what are they going to be looking for as you come with your, diagnostic biomarker test or your treatment or whatever you're developing is to think of how do we get adoption. And if not, then going after, a consumer pay market.
Yeah, exactly. Yeah, or
helps.
even, the consumer pay market as a bit of a bridge while you sort of generate that data. Very common sort of approach, especially with some diagnostics.
Yeah.
So maybe the last question here before we move to the next part. So the question asker is asking, so will early genomics use improve personalized medicine in treatment or prevention? And where do you see this happening first?
Yeah, so I think it will. I mean, I think it's already happening in this certain spaces. So cancer for sure. So the ability to identify individuals at increased risk of, hereditary cancer is occurring. The use of biomarkers and tumor biomarkers to direct treatment. So if you have particular biomarkers in your tumor, directing treatment is happening. So there's tons of work in the oncology space. So precision oncology is definitely going to lead and is leading. In our rare disease space, so those hereditary diseases, definitely leading. Pharmacogenomics is coming along very, very rapidly now. So identifying those variants. I think that the next harder step is going to be in the chronic disease space, right? So what are we going to do with the chronic diseases of adulthood? And those could be neurodegenerative diseases, cardiovascular disease, diabetes. Like, are we going to get biomarkers and the ability to early identify individuals who are at risk, fatty liver disease, and like all the stuff that creates a big burden in the, so there's a lot of push in that space. And in the pediatric space, it's mostly going to be around complex disease as the next space as well, neurodiversity issues and mental health issues. But I think that's your next wave. So it's here definitely in a few really active spaces, and then there's a few kind of rapidly kind of chomping at the heels, which is exciting.
Awesome. Well, fantastic, Francois. That's all the questions we have today, but it was a real pleasure chatting with you and hearing your firsthand experience actually delivering outcomes for patients using these personalized medicine approaches. So again, thanks so much for your time this morning.
Okay, appreciate it. Thanks, everyone.
All right, next up in our program, we'll be hearing from 3 startups leveraging genomic technologies in various capacities. Just a gentle reminder that we'll have a short Q&A after each one. So prepare your questions and put them into the Q&A function down below. And you'll also be seeing a pop-up window in your Zoom, whether you'd like an introduction to that startup for further follow-up, which Plug and Play will facilitate after today's webinar. So our first startup is Dr. Isha Sharma from Lamar Health. Lamar Health is developing a novel software platform for clinical geneticists to save time while improving patient care. Take it away, Isha.
Hi, my name is Isha. I'm CEO and founder of Lamar Health. We're building the Flatiron for rare genetic disease. Flatiron was a company in the cancer space which provided a software service to cancer clinics, and in doing so collected a lot of information on patients and then provided that back to patients, providers, and pharma companies. This is what we aim to do in genetics. I'm super excited about this space because of my own family history in it. My older sibling had an undiagnosed genetic condition, and at 15, she was on several unnecessary medications, which took years of trial and error to figure out. Really, the goal of this company is to make it so that this is not the future for families, so that they get to the best possible care faster. Having spent 10 years in genetics, the other thing that made me really excited about the space was that I saw major market shift happening. Right now, the costs in genetic sequencing are expected to drop by a factor of at least 10x. And this is because the monopoly on genetic sequencing has long been held by one company and their patents are about to expire. I actually had a job offer from one of the competitors that were coming out and saw that potentially the $100 genome was not far off. The other thing that was really exciting was that during COVID, I had the opportunity to work on the mRNA vaccine and saw how these sorts of therapies that could be applied for genetics were extremely tractable and could be used as platform technologies. So that means that you can use them for many different types of genetic conditions. And we see this not just with mRNA therapeutics, but also with CRISPR-based approaches and enzyme replacement therapies. And so really, I wanted to create something where we were at the intersection of the diagnostic side and the therapeutic side, and really being the adapter between these two things. So to do that, I put together the team that I thought would be capable of doing this. It consists of me, PhD in genetics, part of Y Combinator and Creative Destruction Lab. Sergey, who's head of tech, he's been a serial founder and has had various roles both as founding engineer in software as well as ML ops roles and NLP based roles. And really, he does a ton of NLP in his role here. And Aaron and David, who are medical geneticists at leading institutes and have really helped us figure out exactly how to craft something that fits extremely well into their workflow. Our tools help genetic patients and providers get curated, personalized information, and that's driven by NLP. So we offer 3 different things. One is personalized reports on genetic conditions to help patients understand what's going on. Personalized research updates so that they know if new information comes up, for example, if they have an uncertain variant and they want to resolve it. And clinical trial notifications. So if any new treatments or trials come available, they automatically get checked every month and verified if the patient is suitable for that trial. The data we collect and centralize is useful for patients, providers, and life science companies. When I say life science companies, I include both diagnostic and therapeutic companies in that. And the information that we're providing to them are symptom profiles so that they know which conditions are associated with which phenotypes, and that's actually really important when you're developing diagnostics or therapeutics in the space. Key opinion leaders for specific conditions, so this is identifying the doctors that work on particular rare genetic conditions. EHR records with patient consent, which are useful for clinical trials, and market size for rare genetic disease. And this really helps companies decide which conditions to work on. The competitive advantage that we're building is actually the extremely high network effects by creating a tool that's used by doctors and patients. And so the other really important fact is compared to our competitors, what we specialize in is gathering genetic information. And sometimes for a lot of conditions, the specific mutations involved are really important to know in order to precisely identify which patients are amenable for certain therapies or present with certain symptoms. And so we offer that level of granularity. We launched this month at these leading centers and have been working with the doctors at these institutes for many months now to really optimize it for their workflow. If you're a patient, a provider, or a company in the genetic space, we'd love to hear from you. We're building fast and we'd like to learn more about different companies in the space and ways that we can work together and collaborate to build a better future for genetic patients. That's it. Thank you.
Fantastic. Thank you so much for that presentation, Isha. Do we have you on the line here?
Yep. Can you hear me?
Great. Yeah, sure do. So just a couple quick maybe follow-up questions from your presentation there. Would you be able to elaborate a little bit exactly how your platform is using natural language processing and how that's being implemented in like the clinician's workflow?
Yeah, so specifically what the clinician is able to do is build clinical notes really quickly on it. So we pull, basically we have a clinical note builder tool that pulls information from OMIM, GeneReviews, and PubMed. So those are the main clinical databases that they're using. And we use NLP to basically help synthesize information quickly for them and then convert that into a patient-friendly text when they're complete with that note.
Fantastic. Yeah, I can imagine the time being saved in the clinician's workflow there. Maybe another one, you explained sort of the impact on patients presenting with uncertain variants. Could you elaborate a little bit on that, on how your platform is maybe going to help those patients, maybe future patients presenting with those same variants?
Yeah, so as was mentioned earlier, typically the diagnostic yield from a genetic test is about somewhere between 30 and 40%. It kind of differs between specific specialties. So for example, ophthalmology will have close to 100 actually, but in general, if you're just going into a genetic clinic, it's closer to like 30, 40%. And so there's a ton of uncertain variants that are reported, and over time, these get resolved. So basically, things get published in the literature, things get updated in ClinVar, and that's a slow process, but it does happen over time. And so if you want to know right away whether your mutation has been resolved or not, we actually scan through the available literature, scan through ClinVar, and basically let the patient know, hey, there's been a change. And we send an email both to the patient and the provider so that they can schedule a follow-up and discuss the results.
Awesome. Great. Well, thank you so much for joining us today, Isha. Okay. For our next startup, we'll invite Dr. Jeremy Gershko from Embryome. Embryome is bringing modern genomics and data science to those trying to conceive to help overcome the barriers and challenges facing IVF patients today. The floor is yours, Jeremy.
Hi, I'm Jeremy. I'm a co-founder and the CEO of Embryome. We're transforming IVF outcomes using genomics to give parents and clinicians as much certainty as possible that the first embryo they choose for transfer is the one most likely to result in the birth of a healthy baby. We have an unbelievable team of experts alongside us on this journey, experts in medicine and business and genomics, and they have been an unbelievable resource to us as we figure out how to bring this to market. Shockingly, from what you've heard today, and given the importance and the amount of money that people spend on the process, IVF is the only clinical genetics decision today that's being made without exome or whole genome data. In fact, most IVF, if there is any genetic testing done at all, is testing only for aneuploidy. Does the embryo have or not have the right number of chromosomes? Now, that's a reasonable starting point because about 50% of pregnancy loss and IVF failure can be attributed to embryos with the wrong number of chromosomes. But just like every other area of clinical genetics, there's lots of information outside of chromosome number that has a huge impact on decisions. And we already know that at least an additional 35% of pregnancy and IVF outcomes can be attributed to single nucleotide variants, smaller structural variants, indels, and other types of genetic variation that can only be detected through comprehensive sequencing. So our goal is to bring that data to bear on the IVF decision-making process. And there is tremendous clinical and patient demand for this. Even before we built an MVP, we already have 6 MOUs giving us access to more than 30 clinics, market and thought leaders in the US mostly, but also in Canada and Israel, who are excited to bring this to their patients. Now, given that there's this gap and people understand what it is and the value of it, what are we doing that other people aren't, and why are other people not doing this? It's because there's 3 independent technical challenges in bringing this to market. The first is accurately generating whole genome data from a small number of cells. The second is an analytic pipeline that is oriented towards getting successful IVF cycles and pregnancies. And the third is making all this information understandable and usable by patients and clinicians. We develop proprietary pieces along every step of that. And importantly, this allows us to replace all of the existing tests with a single new test that unlocks thousands of new findings without changing the clinical workflow for embryologists or clinicians. That's the beginning of the story, not the end of the story. So better genetics on its own is a $16.5 billion annual market, but putting it at the core of an IVF data platform allows us to improve the whole ecosystem, where combining it with treatment and outcomes data allows us to understand what works and doesn't in the IVF ecosystem, providing better coverage at reduced cost, better experience for patients, and more access to IVF without having to build more infrastructure. All of that turns a huge flywheel that's better for everyone. So to sum up, IVF is the only clinical genetic test that's a decision that's being made without exome or whole genome data. Clinicians and patients want access to this data, and we've proven that with the MOUs that we've signed. We have a proprietary way to analyze the whole genome of each embryo that replaces current PGT and unlocks thousands of new findings without changing clinical workflows. And that genomic data is a critical missing piece in the IVF ecosystem. By integrating it, our outcomes will reduce costs and improve coverage and access. If you're interested in investing or learning more, please feel free to reach out.
Fantastic. Thank you, Jeremy. Do we have you on the line here? Wonderful. Maybe just a quick follow-up question. I mean, IVF is inherently a very expensive process, as you're aware. Do you have early evidence on how implementing Embryome is going to be able to reduce those costs and maybe reduce the number of cycles overall for the process?
Yeah, it's actually pretty straightforward math. The more certainty you can provide on a per transfer basis for embryos, the shorter and less expensive the whole process becomes. So, based on the population data that we have so far for pregnancy loss, our estimate is that instead of the current best-in-class technology that requires between 4 and 5 transfers for patients to have a 95% chance of having a baby, we can bring that down initially between 2 and 3, and we believe by integrating the rest of the medical data that we can get that really as close to certainty on the first transfer as possible. So it's a huge impact, both in terms of the number of transfers required, but also in terms of the amount of time required for patients to know whether a cycle is likely to be successful or not. And that ability to start a second cycle if necessary sooner has a big impact on the overall outcomes and timeline.
Great, thank you. And then maybe one last quick one. What does the sort of the output to the end user look like from the Embryome report? And what does the actionable information from the technician's perspective look like?
Yeah, so I mean, we think about it really, the decision that we're informing is which embryo should you transfer to have the best possible chance of pregnancy. And so the goal of the reporting output is to reach that conclusion. Based on our initial in silico experiments, we think that most cycles should have like a 96-97% chance of having at least one embryo from our classification that falls kind of in a green light category, where we believe it has an excellent chance of resulting in a healthy pregnancy and a healthy baby. So the idea is to help patients identify that and to explain why other embryos don't meet those criteria as simply as possible. And there's a very complicated workflow, obviously, to get through the variant interpretation and reporting, but at a high level, to just provide that answer to patients and clinicians of which embryo has the best chance of a successful pregnancy.
Fantastic. So Jeremy, we just have a question from the audience. We're going to move on, but I encourage you to maybe go into the Q&A and type a response to the questionnaire there. Thanks so much for your time today, Jeremy.
Yeah, thanks, Nat.
All right. So for our last startup today, we have Dr. Peter Nestorov from Scailyte. Scailyte is developing artificial intelligence technology to enable precision medicine and early detection of complex diseases. Take it away, Peter.
Hello, my name is Peter Nestorov. I'm CEO and co-founder of Scailyte. We're a Swiss startup that started in 2017, and we're on a mission to enable precision medicine. The problem that we address is what I call the 90-50 problem. On the one hand, 90% of clinical trials fail, and the reason for this is lack of clinical efficacy for the drugs in development. On the other hand, the drugs that are already on the market, in 90% of cases, or 90% of the drugs that are already on the market, only work for 50% of patients. And this is quite shocking. The reason for this is that 2 patients with the same disease do not respond in the same way to the same drug. And this is because there are differences between individuals, also differences in the environment, and different interactions between what drugs the patients are taking. And our understanding on this needs to improve. And the way to improve this is by looking much deeper molecularly at what's going on the patient level. And also using biomarkers that can really subdivide the patient population into groups that are responding to different treatments. Nowadays, most biomarkers are as the example here, CRP, which is an inflammation biomarker. And if the levels of CRP are high, the clinician understands that there is some inflammation process going on. But it doesn't tell you where this comes from, what is the reason, and also to which treatment the patient would respond in the best way. So the clinician and the patient are stuck in a maze. Now, if we have the situation with new molecular technologies where we can generate a high resolution map and actually get potentially the insight that is needed to take the decision on the right treatment, then we're in a much better situation. But then also that's quite complex. And what Scailyte has developed is an AI-driven platform that takes highly complex and high-dimensional molecular data, such as single-cell data, and then analyzes this in a clinical context by finding patterns, or in other words, cells and genes that are associated with a clinical endpoint. So we can now, by looking at this data, understand which cell types and also which gene expression patterns are relevant to differentiate between responders and non-responders for a certain drug. In other ways, we are allowing the clinician and the patient to find their way in this complex data and know how to reach from point A to point B in the best way. And the process and the workflow for our biomarker discovery approach is described here. As an input, we take different molecular data, among other single cell data and clinical data. We feed it into our platform SkyVision, and then we look for different patterns. Ultimately, these patterns are gene lists, which we can then use to prototype clinical assays. The assays could be either developed further as products to be launched as diagnostics on the market, or to be used in the clinical R&D process. And over the last 5 years, we've raised in total 16 million Swiss francs. Most recently, we closed a Series A with the new investor Mirabaud Bank joining. We also raised money from non-dilutive programs like the InSwiss here in Switzerland. And we've been deploying this money to build an ecosystem for precision medicine, where at the core, we have our SkyVision platform that takes omics data and uses AI to find the biomarkers. And we monetize this in two different ways. One is through offering discovery services to biotech companies and pharma companies, and also building strategic partnerships for the long-term revenue generation. And the other way is also to build a precision diagnostics portfolio. We've been partnering with a number of hospitals here in Switzerland, but also in France, South Korea, and other countries to generate single-cell data in a clinical context and look for biomarkers in different indications with high unmet clinical need. And our most advanced project is in endometriosis, where we're pioneering the discovery and development of molecular diagnostics for this disease that affects 1 in 10 women. And currently there is no molecular diagnostics for endometriosis available. And behind all this, of course, is a great and very diverse team that brings together competence and experience from different fields, from industry, from academia, engineering, biomedical sciences, bioinformatics. We also set up an amazing board of advisors, including our co-founder, Professor Klassen, who is now at the University of Tübingen, and Professor Brenner from the Harvard University. And this is the whole team in front of our office here on the Novartis campus in Basel. And it is driven by a passion for patients, the passion for science, and passion to pioneer. Thank you.
Fantastic. Thank you for the presentation, Peter. Do we have you on the line?
Yes. Hey.
Great. So just a couple questions on Scailyte here. Could you elaborate a little more on how your SkyVision platform overcomes the black box challenge of implementing AI algorithms in precision medicine and discovery programs?
Yeah, I like this question. Thanks for asking. So we don't see our AI as a black box because we control the whole process from inputting the data and knowing what's going on in the processing step, and at the end also having an interpretable result. So also in the explanation that I gave before, what we get is the patterns ultimately are gene signatures. So we know exactly, for example, which cell types are related to a clinical endpoint, and also which genes are contributing to this association with a clinical endpoint. So it's not a black box, but it's actually giving us a quite precise answer on which genes could be used as biomarkers.
Great. And then, thinking about your lead diagnostic product in endometriosis, given that there's no, wide available molecular diagnostics at all for this disease, could you describe what kind of an impact this tool might have on the care pathway for these women?
For endometriosis, we started this project a few years ago because we identified this really as a big unmet need. And the impact that we imagine our diagnostics to have in, let's say, 2 years when we hopefully successfully launch it on the market would be first to shorten the time to diagnosis, because currently it really takes a very long time, up to 7 years here in Switzerland and also in the US to diagnose these patients. And it also requires an invasive approach. So currently the only way to do this is through laparoscopy. And we're developing a molecular test which will be non-invasive and will be able to shorten this diagnostic journey down to a few months instead of years.
Great. Well, thank you so much for joining us today, Peter.
Thanks a lot.
All right. So that concludes the formal part of our webinar here. I want to say again a big thank you to all of our wonderful speakers. It's sufficient to say there's an immense amount of hope left, I think, in the story of precision medicine, a story that's more of an evolution in the way we do medicine rather than an overnight revolution. But thank you to everyone for joining us today and attending. Before we adjourn, you'll see a quick little pop-up in your Zoom window. We kindly ask that you share your thoughts on today's webinar as Plug and Play continues to provide the community with relevant and insightful comment. So thank you so much and have a wonderful Wednesday.